morning to all the students. So, today we are going to deal with the concept fungi from our 100 days concept, okay, for NEET 2026. So, here in this fungi, definitely in every alternate year we will be getting one question from fungi. That also major important part is with respect to classification and reproduction, okay, classification and reproduction of fungi. Fine, ma'am. So, first let us look into the basic general characteristics of fungi and then its classification and then its reproduction in detail. So, let us get started first with general characteristics. So, fungi is a unique kingdom with eukaryotic, thalloid and achlorophyllous. They are purely heterotrophic mode of nutrition and heterotrophic organisms. Who introduced the kingdom fungi in which kingdom system of classification? This can be an objective. So, who introduced the kingdom fungi? R.H. Whittaker. Okay, R.H. Whittaker has introduced the kingdom fungi in his 5 kingdom system of classification. Okay, in his 5 kingdom system of classification. And how can you describe fungi as? There is no organ or organ system formation in fungi. Okay, they are simple thalloid, eukaryotic, purely or strictly heterotrophic organisms. Next, they show great diversity in morphology and habitat. If the question is with respect to metabolic diversity, which kingdom is highly metabolically diverse? If that is the question, then our answer should be Monera. If the question is great diversity in morphology and habitat, then our answer should be fungi. So, here fungi can vary from unicellular yeast to complex edible mushrooms or toadstools. Remember that. Next, fungi are cosmopolitan in distribution. They are present everywhere, mostly in the warm and humid places. Next. Yeah. So, hyphae. What are hyphae? Thin filamentous, long, slender, thread-like structures. Group of this hyphal strands will form what? Mycelium. So, this mycelium also can be septate or aseptate. If the mycelium or the hyphal strands are aseptate, that will turn out to be cenocytic or multinucleated condition. They show cenocytic or multinucleated condition, which are continuous tubes. So, this aseptate condition is observed only in one class, that is Phycomycetes. Remember beta, that is very important. Next, septate mycelium. So, septate mycelium, if you look into, again bunch of hyphal strands, wherein this hyphal strands, they are septate, which are uninucleated condition, not multinucleated over here. So, here this septa based on the pores or the septal pores, we have 2 different types. Okay, in Ascomycetes you can clearly see pore like septa, okay, simple pore like septa, whereas in Basidiomycetes a barrel shaped structure is observed like this, which is called as dolipore septa. Remember, that is a characteristic feature both for Ascomycetes and Basidiomycetes. So, septate type of mycelium is observed in Ascomycetes, Basidiomycetes and Deuteromycetes. Firstly, Ascomycetes, here it can be pore like septa. Basidiomycetes will show dolipore septa, which shows like barrel like structure, and then Deuteromycetes is simple again pore like septa, okay, normal septate condition. But then dolipore septa is observed in Basidiomycetes, pore like septa is observed in Actinomycetes. Clear, right, ma'am? Next question. Cell wall. Okay, cell wall in fungi is made up of chitin. Chitin along with polysaccharides. So, it is also called as fungal cellulose. Okay, so cell wall is fungal cellulose. Next, nutrition. Nutrition, 3 modes of nutrition we can observe: saprophytic, parasitic and symbiotic. How these 3 modes of nutrition is operated? Let us look into that keenly. Saprophytic nutrition, where it is completely relayed on dead and decaying substances. So, this is completely relayed on dead and decaying substances. Dead and decaying substances. So here in saprophytic mode of nutrition, what happens is the fungal mycelium or this hyphal strands, they will be releasing out the enzymes. Okay, enzymes will be released out to the dead and decaying matter which is in close proximity to this. Okay, after this, once the enzymes are released and acting upon this, they will be turned to simpler substances. Complex matter turns to simpler substances, and this simpler substances will be released to the matter again. Okay, so this will be absorbed by this fungal hyphae or the fungal body. Okay, so how saprophytic mode of nutrition is operated, their target is the dead and decaying matter. They will be releasing out the enzymes which solubilize the complex matter into the simpler substances, and they will be absorbed again back to the hyphae. Parasitic mode of nutrition is relying on the living plants and animals. Parasitic mode of nutrition. Symbiotic mode of nutrition, they are in mutual association or symbiotic association with the organisms, like for example with algae or roots of higher plants. Fungi along with algae, fungi in mutual association with algae will form what? Lichens. Fungi along with symbiotic association with algae forms lichens, which are pioneers of plant succession. Okay, and they are the best pollution indicators. This is one symbiotic association. Second is with roots of higher plants. Best example is pinus. Pinus roots, they are associated with fungi. They are associated with fungi, and you call it as mycorrhizal roots. Okay, mycorrhizal roots, wherein fungal association with the roots of pinus will increase its absorption capacity. They will absorb more water and nutrients. Clear, right? Next, reserve food. Okay, reserve food is what? Glycogen and oil. Glycogen and oil are the reserve food materials of the fungi. Clear with the general characteristics right now, beta? Next, reproduction. Let us look into the basic framework of reproduction over here. Vegetative mode, asexual mode and sexual mode. Vegetative mode is of three types: fragmentation, binary fission, which is also called as amitosis, and budding. Okay, asexual mode of reproduction is through spores. What are these spores? Asexual spores. Asexual spores. They can be conidia, sporangiospores, zoospores and aplanospores. Aplanospores. Zoospores here are motile, aplanospores are non-motile. Clear, right, ma'am? Next, sexual spores. Sexual spores can be oospores, ascospores and basidiospores. Till here it is clear, right, na beta? Next, let us look into vegetative reproduction. Fragmentation, where the parental hyphae, parental or matured hyphae will be fragmented into small hyphal strands. Second is budding. Okay, in budding, from the parental bud, small buds will be arising over here. Okay, this, when detached from the parental body, they fall onto the substrata and grow into a new hyphae. This is about budding. Binary fission, which is also called as amitosis, where simple cell division without any involvement of spindle apparatus will be taking place in binary fission. So these are the three modes of vegetative reproduction. Coming to asexual and sexual reproduction, first let us have a brief idea on this. Okay, so yeah, look over here. First, asexual reproduction we will be looking into. We know that fungal body is haploid. Okay, fungal body is haploid, do remember that. Okay, so this haploid fungal body will be giving rise to either sporangium or any such bodies over it. Okay, so over this fungal mycelium, sporangium might be developed, and from this small asexual spores will be released. Through sporangium, sporangiospores, conidia are the exogenous spores. Okay, so conidiophores or sporangiophores will be developed over the mycelium. They are again haploid and they undergo mitosis, will be releasing out the spores. These spores falling onto the suitable substrata, they will germinate and they will grow into a new fungal mycelium body again. So, fungal mycelium will be developing what? The fruiting bodies like sporangiophore or conidiophore. Sporangiophore and conidiophore will be releasing out sporangiospores and sporangiospores and conidia. Clear, right, ma? So, over this mycelium, sporangium and conidiophores will be formed, which are haploid. They will be releasing out sporangiospores and conidia. They, on falling onto the suitable substrata, they will be germinating into the new mycelium. So, only mitosis will be involved over the asexual reproduction. In sexual reproduction, 3 stages will be there. Okay. So, you will be coming across 3 stages. First is plasmogamy. Second will be karyogamy, and third will be meiosis. Okay. So, you have plasmogamy, karyogamy and meiosis. Plasmogamy is nothing but protoplasmic fusion. Okay. Protoplasmic fusion in between 2 compatible hyphal strands. Okay. That will be resulting in heterokaryotic stage. Okay. The fusion between 2 compatible hyphal strands will result in heterokaryotic structure, and this is what? Karyogamy. Okay. That results in formation of zygote, and zygote will be diploid over here. Okay. This, after karyogamy, in ascomycetes and basidiomycetes, dikaryon structure is observed. Okay. Dikaryon condition is observed. Dikaryon condition, that is N plus N condition is observed, which later forms diploid condition or zygote, or you can say diploid ascus in ascomycetes, diploid basidium in basidiomycetes. That will further undergo reductional division or meiosis, again forms haploid spores. This haploid spores falling onto the suitable substrata, they will be growing into new hyphae. So, here one key point you have to remember beta, what is that? In asexual reproduction, purely mitosis happens, there is nowhere where meiosis is taking place over here, and here what are the bodies, fruiting bodies that are forming over here? Those are sporangiophores and conidiophores, and they will be releasing out sporangia and conidia, right. And in sexual reproduction, 3 specific stages. One is plasmogamy, karyogamy and meiosis. Plasmogamy, wherein protoplasmic fusion happens between 2 compatible hyphal strands, then this leads to heterokaryotic condition, and then karyogamy, dikaryon stage is observed in ascomycetes and basidiomycetes. This results into diploid zygote and diploid structure, ascus in ascomycetes, basidium in basidiomycetes. Further reductional division over there results into haploid spores. This haploid spores falling onto suitable substrata develops into haploid mycelium. Till here it is clear, na beta? Next, let us look into the asexual spores and sexual spore in detail. This slide is very much important. Most of the times you got questions only on the spores of asexual spores and sexual spores. Okay. So, look into that keenly, with keen concentration listen, take screenshot of this slide if you need. Asexual reproduction and sexual reproduction. Asexual spores are what? Zoospores, conidia and sporangiospores. Just add on aplanospores here also. Here just add on aplanospores. So, here zoospores are motile and biflagellate beta. Aplanospores are non-motile. Aplanospores are non-motile. Conidia are non-motile and exogenously produced. Sporangiospores, they are thin-walled, non-motile, endogenously produced. Thin-walled, non-motile, endogenously produced from the sac, what? Sporangium. Clear, right, ma? So, conidia are released from the tips of the conidiophores. So, these are the 4 spores of asexual reproduction. Sexual reproduction, you have oospores, ascospores, basidiospores. Oospores can be non-motile and endogenous, ascospores non-motile, endogenous, and they are from what fruiting bodies? Ascocarps. Ascocarps are haploid only. They will bear haploid ascospores. Basidiospores are again non-motile, exogenous. They are produced from the haploid basidiocarps. Till here it is clear, right ma? Asexual reproduction and sexual reproduction. Here one of the sexual spore you can add it, that is zygospore from zygomycetes. These are also endogenously produced. Endogenously produced. So you can take a screenshot of this. This is very important. Many at times the questions are directly asked on the spores only. One small trick, that in asexual spores, in amongst all the asexual spores, the only exogenously released asexual spore is conidia. Amongst all sexual spores, the only exogenously released sexual spore is basidiospores. Till here it is clear, right ma? Next, based on the morphology of mycelium, mode of the spore formation and fruiting bodies, on what basis fungal classification is done. Take a keynote of all these three statements. Based on what fungal classification is done? Based on morphology, based on mode of spore formation, and based on fruiting bodies. They are classified into 4 classes. First is Phycomycetes, second Ascomycetes, Basidiomycetes and Deuteromycetes. Phycomycetes, they again have oomycetes and zygomycetes based on their sexual spore. Here in oomycetes it is oospore, zygomycetes it is zygospore. Okay. Next, let us look into each class in more detailed way. Phycomycetes is also called as algal fungi, important. Okay. Phycomycetes is also called as algal fungi, and this is the primitive fungi actually found in aquatic habitats on decaying wood, and they are found as obligate parasites. Mycelium, if you have to see, that is aseptate or cenocytic condition or cenocytic mycelium. Okay. Asexual reproduction is through zoospores and aplanospores. Okay. Here amongst these two, which is motile ma? Zoospores are motile, aplanospores are non-motile. Sexual reproduction, if you have to look into, fusion of 2 gametes. So this fusion can be either through isogamy, anisogamy and oogamy. What is isogamy? If both the gametes are similar in structure and physiology, they are fusing, so it is called as isogamy. If 2 gametes or 2 compatible strains which are fusing, they are differentiated either through physiology or through structure, then it is anisogamy. Small motile male gamete fuses with large non-motile female gamete is called as oogamy. So sexual reproduction can be any of these 3 types: isogamy, anisogamy or oogamy. Okay, here the concept is about fusion of 2 gametes. It can be of any of these 3 types. Products after any of these 3 types of sexual reproduction: in oomycetes, oospore is formed; in zygomycetes, zygospore is formed. Okay, oomycetes, oospore is formed, and zygomycetes, zygospore is formed. Here you can see oospore germination, right, and this is zygospore. See, compatible strains after mating, they are forming what? Zygospore in zygomycetes. So this will be the site of meiosis. Nowhere in Phycomycetes you can see that 3 stages of plasmogamy, dikaryon stage and meiosis. Dikaryon stage is not present in Phycomycetes. Remember that ma. So there is no dikaryon stage over here. Here oospore and zygospore, which are the products of the sexual reproduction, this will be the site of or the location of meiosis, and spores will be formed which will germinate into new hyphae. The spores are haploid and new hyphae also will be haploid. Clear with the sexual reproduction of Phycomycetes, right ma? So here if you look into the examples, okay, examples you can see Mucor rhizopus. Rhizopus is also called as bread mold. Okay, albugo, where in albugo this shows parasitic mode of nutrition, right? Albugo shows parasitic mode of nutrition. mode of nutrition, okay, which gives you white spots on the mustard leaves. So, the best examples of phycomycetes: mucor, rhizopus which is also called as bread mound, and albugo, which shows parasitic mode of nutrition, which gives white spots on the mustard leaves. This shows parasitic mode of nutrition. So, the examples how you can remember: RAM, okay, rhizopus, albugo, mucor, wherein albugo comes under oomycetes, rhizopus and mucor under zygomycetes. Clear, right? Next, ascomycetes. Ascomycetes shows great variation in morphology, where this shows highly variety of species from unicellular yeast Saccharomyces cerevisiae to multicellular penicillium, claviceps and all. Okay. So, this will be the diversity in its morphology, ascomycetes. These are saprophytic, decomposers, parasitic and coprophilous as well. Mycelium in ascomycetes is septate, and this shows pore-like septa, as we have already discussed in the general characteristics. Mycelia will show what type of septa in ascomycetes, ma? Pore-like septa. Clear, right? Asexual reproduction, if you have to look into, special hyphae or conidiophores will be formed. So, from the basic hyphae, conidiophore will be originating like this, conidiophore with foot cell, and at the above these structures are called as sterigmata. Sterigmata will be bearing the conidia. Through the sterigmata, conidia are exogenously exposed and they are released exogenously. So, this is about conidia. Conidia are the asexual spores in ascomycetes. Do remember in each and every class what are the asexual spores and what are the sexual spores. So, here the special hyphae over here is what? Conidiophores. They produce conidia exogenously. Conidia on germination they will give rise to mycelium. So, here two additional points that you have to remember is foot cell is the connecting dot from this hyphae to the conidiophore, and at the above you have sterigmata which is releasing out the non-motile asexual spore, that is conidia. Clear, right, ma? Now, sexual reproduction. So, here this in sexual reproduction fruiting bodies will be formed. They show different sac-like structures. So, those saucer shaped, saucer or cup shaped apothecium, flask shaped perithecium, clistothecium which is globose shaped. Okay. So, wherein here ascocarpus haploid which has both antheridium and archegonium, male and female gametes, okay, where they will fuse and form ascus, ascus which is diploid. Okay. So, inside ascocarp fusion happens, and inside ascocarp only ascus is formed. Ascus is diploid. Now, this is the site of meiosis. After meiosis, this will produce 4 spores. Okay. These spores are haploid. Okay. Haploid spores falling onto the suitable substrata, they will germinate into a new hyphae. Clear, right, ma? So, here what is the site of meiosis? Ascus. And where is the fusion happening? Inside the ascocarp. Based on their shape, we have 3 types: apothecium, perithecium and clistothecium. So, till here it is clear, right, ma? Next, examples of ascomycetes. I can give you a mnemonic over here, that is ACP and T. This is the best mnemonic where you can remember all the examples of ascomycetes. A for aspergillus here, C is claviceps, penicillium, neurospora, T truffles, morels and truffles, and here Y is for yeast. You can remember easily, Navita. You can take down this mnemonic as well, ACP and T. Okay. Aspergillus, claviceps, penicillium, neurospora, morels and truffles, and then yeast. Clear, right? And then third class, which is the most advanced class of fungi, that is Basidiomycetes, which is commonly called as club fungi. Okay. So, most well known forms of Basidiomycetes are mushrooms, puffballs or bracket fungi. So, here They grow in soil or on logs or on tree stumps or in living bodies as a parasite. So, this is about habitat. Mycelium is septate and branched only. Okay, mycelium is septate and branched. And septa are of what type of septa? They show barrel type of septa like this, right? So, they show barrel type of septa like this. This barrel type of septa are called as dollipore septa. Dollipore septa. Clear, right, ma? Now, if you have to look into reproduction, we have vegetative, asexual and sexual reproduction. In Basidiomycetes, asexual reproduction will be absent. Vegetative mode of reproduction is again fragmentation. Fragmentation, budding, fission, those are vegetative modes of reproduction. Asexual reproduction is absent in Basidiomycetes. Sexual reproduction is only through basidiospores. Basidiospores. These basidiospores are the only exogenously produced sexual spores. Okay. So, the only mode of reproduction is sexual, that is through basidiospores, which are exogenously produced, and vegetative modes are common again: fragmentation, fission and budding. So, here sexual reproduction, if you see, ma, basidiocarps. So, this is what over the hyphal strands basidiocarp is formed. This is the fruiting body. Okay, fruiting body basidiocarp, which is haploid. Basidiocarp, which is haploid. Here inside the basidiocarp, fusion takes place between male and female gametes, and after that this results in the formation of basidia. Okay, this basidia is diploid. This will be the site of meiosis, meiosis or reductional division, and this results in the formation of 4 spores. Spores will be released out, and basidiospores will be released exogenously like this over the fruiting bodies. This is the basidium, which is the site of, just a second. So, this is the basidium, which is the site of meiosis, will be releasing out 4 spores. Okay, those are basidiospores which are released and falling onto the suitable substrata. They will be growing into the new hyphae or new mycelium. Till here it is clear, right, beta? Next, fructification is large and conspicuous in Basidiomycetes, as it is the advanced class of fungi. Now, this is the most advanced class of the fungi. Now, examples: Agaricus, Eustylago, Puccinia, Polyporus and Lycoperdon. Agaricus best type is mushroom. Eustylago, which is a smut fungi. Puccinia is rust fungus or rust fungi. Polyporus and Lycoperdon. Polyporus is a bracket fungi. Lycoperdon are also called as puff balls. Remember the synonyms, okay, very important. Fine, ma. Next, last class of the fungi, that is Deuteromycetes. Deuteromycetes is called as fungi imperfecti or imperfect fungi, wherein the mycelium is septate and branched. Mycelium is septate and branched. Fine. These are largely decomposers. Few of them are saprophytic and parasitic, but largely they rely on what type of nutrition? Decomposers. They litter, and as most of them are decomposers, they play a major role in mineral cycling, and their only mode of reproduction is asexual reproduction, that is through conidia again. So, conidia are asexual spores both in Ascomycetes and in Deuteromycetes. While establishment of the linkages, so there is one paragraph in our NCERT which clearly says that most of the examples which are dropped in Deuteromycetes in the beginning of classification, where they again placed into Ascomycetes and Basidiomycetes if they are conducting sexual reproduction. Okay. So, all those fungi which are placed in Deuteromycetes initially. Once after clearly analyzing that they are conducting sexual reproduction, they are moved again to Ascomycetes and Basidiomycetes. That is the gist of that paragraph actually, but look into this as well. While establishment of linkages, fungi with perfect sexual stages are moved to Ascomycetes and Basidiomycetes. Clear, right ma? So, how can you remember the examples like cat? Simple, right? The best mnemonic for Deuteromycetes to remember is cat, Coletotrichum, Alternaria and Trichoderma. Okay, now. So, this is the final table. Okay. Good afternoon. You can answer this. Okay. Phycomycetes is also called as algal fungi, right ma? This is also called as algal fungi. Okay. Ascomycetes is also called as sac fungi. Basidiomycetes most commonly called as puff balls or bracket fungi. Bracket fungi. Deuteromycetes is called as fungi imperfecti. Clear, right ma? And mycelium, here if you have to look into Phycomycetes, this is aseptate and cenocytic. Fine. Fungal mycelium is aseptate and cenocytic. Ascomycetes, this is septate. Ascomycetes is septate and it has what type of septa? Pore like septa. Basidiomycetes is also septate and branched. Septate and branched with dolypore septa. Dolypore septa. Fine. And Deuteromycetes, this is also septate and branched. Fine ma. Asexual spores and sexual spores, highly important. Okay, we can expect a direct questionnaire just from asexual spores and sexual spores. Okay, so in Phycomycetes, what are the asexual spores that we have come across? Sporangiospores, sporangiosspores, zoospores and aplanospores. Zoospores and aplanospores. In Ascomycetes, what are the types of spores we have come across? Sporangiospores and conidia are the asexual spores. Basidiomycetes will not show any asexual mode of reproduction. Deuteromycetes again conidia is the asexual mode of reproduction. Now sexual spores in Phycomycetes, it can be oospores or zygospores. Ascomycetes, ascospores, which are endogenously produced from ascocarp. Basidiomycetes, basidiospores, which are exogenously produced from basidiocarps. Deuteromycetes, sexual reproduction is absent. Lastly, when it comes to examples, just recall the mnemonics which I gave, it will be easy for you to answer. Phycomycetes, it is RAM, ram, wherein Rhizopus, albugo and mucor, easy right. Ascomycetes, ACP and team. Aspergillus, claviceps, penicillium, neurospora, which is highly used in genetical work, and morels and truffles, and then yeast or Saccharomyces cerevisiae, which is unicellular fungi. Basidiomycetes I didn't give you previously, right, take down, that is a pulp. Okay, Agaricus, Polyporus, Eustylago, Lycoperdon, Puccinia. Okay, that is a pulp. Next, Deuteromycetes, symbol mnemonic again, that is cat. Coletotrichum, Alternaria and Trichoderma. Clear, right? So, this is all about fungi, classification, classes, examples, most importantly reproduction part, that is asexual and sexual. Highly focus and keep additional attention to remember the asexual spores, sexual spores and examples of all the classes of fungi. With this, your preparation for NEET with respect to fungal concept is done. Thank you.